would be variable expressivity and Turner syndrome. I'm Chris Lee.
I'm Ruben Andre. I'm Stephanie Ebing. East McKenzie.
So a little bit of introduction about Turner syndrome is our introduction question first and foremost is what accounts for the phenotypic heterogeneity and variable expressivity in Turner syndrome patients. So Turner syndrome is a genetic condition that only affects girls, and it is not inherited from parents, nor it is significantly influenced by environment. It is the all-maternal age increases, non-disjunction, as well as the monosomy for the X chromosome can arise either by failure to transmit a sex chromosome to one of the gametes, or by loss of sex chromosome from the zygote or early embryo. There's also been identified different variable expressivity in different patients in different Turner syndrome patients.
So as we said, there are two forms of Turner syndrome, or two ways in which we can characterize Turner syndrome. So there's true monosomy and there's also mosaicism. So in true monosomy, this occurs in non-disjunction during meiosis of the gametes. So in non-disjunction, one of the gametes either have only one X chromosome or a gamete only doesn't have any X chromosome. So when this gamete is fertilized, it only has one X chromosome, and this is known as true monosomy. So all the cells in that person that will develop have a karyotype of 45X. However, Turner syndrome can also occur after fertilization during zygotic development. And this occurs during non-disjunction in mitosis of the fetal cells. And this results in the same outcome where you have a cell or you have a line of cells that only have one copy of the X chromosome, while you have other cells that have 2 copies, 3 copies, and so on of the X chromosome.
So symptoms for patients who have Turner syndrome, one is that they have a short stature, and more than 90% of the patients that have Turner syndrome have some type of ovarian dysgenesis, which is basically they have a problem with their basically fertility. And then they also have physical abnormalities such as broad neck and broad chest. So this flow chart that we have here is a molecular mechanism for of Turner syndrome. So the most important information we highlighted here is gene dosage effect, and gene dosage effect can have a loss of the show X gene, which is basically the short stature gene, and as well as the loss of Y growth gene. So these two genes can affect short stature and skeletal stigmata.
So the research paper that we've chosen or was given to us to present on, the title of it is Gene Dosage as a relevant mechanism for the determination of ovarian function in Turner syndrome patients. So Turner syndrome is quite prevalent. So as we know, it only affects females. So 1 in every 2,500 to 1 in every 3,000 females are born with Turner syndrome. So as we previously said, Turner syndrome can be characterized by true monosomy, so where the karyotype is 45X, or it can be characterized by mosaicism, where the karyotype is 45X. Some cells are 45X, while other cells are euploid, which are 45XX. So there can also be chromosomal, there cannot be chromosomal abnormalities in the euploid cell lines. So as we also discussed, short stature and ovarian dysfunction are two characteristic phenotypes of Turner syndrome. So the ovarian phenotype comes from ovarian dysgenesis. So dysgenesis refers to an organ not developing properly in the embryonic stage. And because in this case the ovaries were not properly developed in the fetal stage, this results in the patients not having complete pubertal development, or there's no pubertal development. So there are several ways, or conventionally we use karyotypes to identify the chromosomes, to look at chromosomes are present, and this is a typical way of seeing if a person has Turner syndrome or not, because if they have Turner syndrome, then we know that one chromosome will be lost, or one of the X chromosomes would be lost. However, there are limitations to karyotyping as it cannot detect copy number variations. So this is where there's a duplication or there's a deletion in some parts of some of the genes in the X chromosome, and therefore this is why conventional karyotyping is not like shows a whole picture of whether a person has Turner syndrome or not. So two main categories of Turner syndrome patients that are pointed out in this research paper are spontaneous menarche patients and primary amenorrhea patients. So spontaneous menarche refers to, well, the term itself refers to the first menstrual cycle that a female has. That's why it's called spontaneous. And this can occur anytime before the age of 16. So there are some Turner syndrome patients who do have normal pubertal development and have spontaneous menarche. Therefore, they're categorized as spontaneous menarche patients. However, there are some Turner syndrome patients that have infertility issues, which is characteristics of Turner syndrome, and these patients don't have normal pubertal development before the age of 16. So the study question from this paper is, can different molecular techniques be used to identify molecular signatures in Turner syndrome patients with spontaneous menarche?
So as a group, we have come up with different research questions according to the paper, and they are: is there copy number variations in the genome of Turner syndrome patients with primary amenorrhea and spontaneous menarche? And what are the structural characterization of these copy number variants? And is X chromosome mosaicism present in SMPA patients? And could the occurrence of spontaneous menarche be associated with mosaicism?
Okay, so the first experimental design of this paper set out to find out if there is copy number variation in the genome of Turner syndrome patients with primary amenorrhea and spontaneous menarche. So the first thing they did was to collect whole blood samples from all 40 Turner syndrome patients that were included in the study, and also collect whole blood samples from 10 LT donor patients. 10 LT donor persons. And from this whole blood samples, the genomic DNA was extracted. So the method used to analyze whether copy number variance is present or not is a method called array comparative genomic hybridization. And this is a technique where so as we said, we have genomic DNA from a patient and from a reference. So the reference, as I will continue mentioning, is the refers to the DNA or the genomic DNA from the healthy donors. So what happens is that these genomic DNA, they're cut into small fragments, and then they are co-hybridized. So as we know in hybridization, we can have hybridization with a DNA strand, RNA strand, that complementary base pairs. So this is basically what happens between the fragment of DNA from the patient and the fragment of DNA from the reference sample. And these are basically co-hybridized on an array and analyzed whether there is a difference between the patient genomic DNA and the reference genomic DNA. And these are done in small DNA fragments until the entire genome of the patients is analyzed. So the output of this is or what they do with the data from this. They use the software and they actually do a log ratio plot. So what a log ratio plot shows is that, is there any difference between the genomic DNA of the patient and that of the reference? If there is no difference, we should have like let's say a thread line that comes straight down that shows no difference. So if you would notice, like for example here, most of the line, if you draw a line straight down, you would see that there's kind of no difference with the black dots here. However, when there is a difference in the genomic DNA of the patient and that of the reference, you do see a shift from your mean of the 0. So if there's a shift in the positive direction on the log scale, this indicates that there's a duplication in the patient's gene, or if there's a shift to the left in the negative direction, that indicates that there is a deletion in the patient's genome. So they did all of this with the genomic DNA from the Turner syndrome patients, and what they did was to identify the genes that there are copy number variants present in, and also characterize these copy number variants as either rare or common. So common copy number variants are those copy number variants that are present in both the reference sample and the patient sample, and these were not characterized as significant since they were present in both. However, rare copy number variants are those that were present in the patient sample, and it was only present, let's say, maybe 1, 2 patients, or so on. And then after that, they compared the copy number variants between the primary amenorrhea and spontaneous menarche patients. So presented here is a logarithm plot taken from two different patients. So we have, or from 3 different patients. So we have patient PA5 represents primary amenorrhea patient number 5, SN1 represents spontaneous menarche patient number 1. So as we said, ACGH, array comparative genomic hybridization, detects whether there's a deletion or duplication in genes. So what we saw in our primary amenorrhea patients is that there was a deletion in in our first primary amenorrhea patient here, we saw there is a deletion in the papa gene. So the papa gene is a pregnancy associated protein gene, and this gene is very important in pregnancy and development of the follicle. And in patient number 4, we saw that there was a duplication in the PDE8A gene, and this gene is involved in the ovary development. And also in this same patient, we also saw a deletion in the PRKX gene, which is a cyclic AMP-dependent gene on the X chromosome. While in our SM patient, we saw a duplication in the BMP15 gene, and BMP15 gene is the gene for bone morphogenic protein, and this gene is very important in ovarian maturation and follicle development.
So the second experimental design was based on the second research question, and overall we're trying to detect mosaicism. So first of all, what the researchers did was they collected buccal-mucosal cells from 13 Turner syndrome patients. Out of the 13, it was 6 spontaneous menarche and 7 primary amenorrhea patients, and then the rest were healthy patients and included males included as well. The cells were fixed, and then they looked at proper cell morphology. So the first technique they did was iFish. So we know fluorescent in situ hybridization, but however this one is iFish. So they look at the interphase level of the cells, and once they performed that, they basically fluorescent the genes, they probe the genes, and they probe for BMP15, which is important for oocyte maturation and follicle development. And then the next technique was RT-PCR, and that's real-time polymerase chain reaction. So as we know from back in general genetics, we use the polymerase chain reaction to basically amplify the DNA and to see basically the amount of DNA we have. And in this case, they probed the genes GYG2 and PRKX. And GYG2 is really good for X-linked situations, and then PRKX is good for basically sex reversal disorders, so it's really important for Turner syndrome as well. And then also they probe the papa gene, which is important for female fertility. And once they did both of the techniques, they pretty much found what mosaicism is, which is the number of, basically they looked at the number of probes, and they were determining the number of probes that they were detected was the number of chromosomes that the patients had. So here is the results. So as stated before, they probed the genes. As you can see here, red is for BMP15, green is for the locus D6ZI, and then you see television probes. So television probes, they're important for basically detecting the arms and the chromosomes. So for example, television 1P probe detects the first chromosome number 1 and the P arm of that chromosome as well. So you can see here in figures 2C that they detected these 2 chromosomes, and as stated previously, they used the probes to detect the number of chromosomes. So in this case, you see that in this figure 2C, you have an unequal amount of chromosomes. So you have 2 you have 2 chromosomes with the same probes, but however, you don't have the pairing of this one. And
How do we know which probe is which?
oh, based on the color. So, sorry, television 1Q probe is red, so you can see it here. And then television XPQ, so that will be on the X chromosome and on the P arm, and then Y would be the Y chromosome on the P arm, and that's the yellow, so you can see the yellow here, and also the yellow as well. And then the same thing goes for the probes red and green. So if you look up here, that's detecting, the red one's detecting BP15, and then green is detecting the locus. So you can see that there's 2 greens, however, there's only 1 in this cell, and then there's 2 green here and 2 red. So that shows there's an unequal number of chromosomes. So that's what the iFish detected. So based on the PCR, RT-PCR, so based on the RT-PCR detections, they basically compared the PA patients and the control, and they found that in your PA4, looking at the PRKX gene and the GYG2 gene, you have your the PA patient has 1 N and the control has 2 N. So that shows you that ideally you're supposed to have 2 N, but however, in this case, your primary memory patient only has 1 N. And then in this section right here, PA4, when looking at the gene PDE8, your PA patient, however, has 3 N, and your control has 2 N. And then lastly, looking at PA5, looking at the Papa gene that was probed, your PA patient has 1 N, whereas your control has 2 N. So with that, with the RT experiment, there was a duplication in the PD8A gene. So that's why here there's more. This patient has more chromosomes than usual compared to the control. And then it also detected deletion in the Papa gene, which was probed. So that's why your primary amenorrhea patient has 1 N except for 2 N.
For the experimental design, we can use the RT-PCR, and the patient is X chromosome is X and present in SMNPA patient. For this design, they had used the buccal and vaginal epithelial cells, and the genomic DNA from these patients were used as a target templates for the detection of disease. And these templates were used as the primers in the RT-PCR, and the controls were 46XX females and 46XY males. And a difference between the PCR and the qPCR is that the PCR is amplification of the DNA towards the completion of the reaction, while the qPCR is the amplification of the DNA as the reaction progresses. And it also uses fluorescent dyes for the identification. And the instrument that we use is 7900 HT-BL1 PCR, and the reference probe is RNAs. And this is a 2-step procedure where the cDNA is made first, and then qPCR is done later using the fluorescent dyes. Yeah, and then the genes that were specified were GY-G2, BMP15, PDE8A, as mentioned earlier, and the Papa gene, and the SMARCA1. And based on the results of from the PCR, the increased fluorescence indicates the increase in the cycles and an indication of the amplified DNA. And this can be put into a standard curve, which shows that the cycle numbers increase indicate the increase in the DNA as the fluorescence increases. And the threshold line is something where the DNA is equal and the same in all the samples. And the intersection of the threshold line and the samples between samples is something where the cycle quantification is known. And putting into a log concentration and CQ graph, we can know that the log concentration of the DNA increases, which indicates an increase in the DNA. As the cycles decrease, which indicates that the lesser cycles you can produce more. The more DNA is amplified. And the results for the experiment included eye-fish analysis in the peripheral blood cells and the deeper mucosal cells of the treated patient. And the green curve, the green signal indicates the locus DXZI on chromosome X, and then blue signal indicates the D18ZI on chromosome 18. And the mosaic condition is known by the signaling of different signals, different number of signals. And as indicated with the yellow arrows, the cell has one signal, one green signal, and two blue signals, which indicate mosaic condition. And this can be confirmed by more PCR data. Gene was specified as GYG2, and this is glycogen. This is associated with the glycogen synthesis, and it's an X-linked gene. And as in the bar graphs, in the SM patients, the spontaneous monarchy patients have 2 copies of the gene in SM4, SM5, and SM6, while the SM1, SM2, and SM3 patients have only 1 copy of the gene. And these were between peripheral blood cells, fecal mucosa, and vaginal cells, while the SM4, SM5, SM6 were only compared for peripheral blood cells. In the PA patients, my primary amenorrhea, there is only 1 copy of the gene in all of the PA patients. And the next specified gene is BMP15, which is a bone morphogenic gene protein, and reflects in this gene called ovarian dystenesis, and this is also associated with ovarian failure. And gene copies identified were in SM1 patients. There's two copies of the gene, and 2 copies in SM4, SM5, and SM6, while the SM2 and SM3 have only 1 copy of the gene. And the PA, there's an observation where PA patient with fecal mucosal cells had 2 copies of the gene, while peripheral blood cells, there's only 1 copy of the gene. And the SMARCA1 gene, it's on the XQ arm, and this is associated with the chromatin remodeling, and this is correlated with cell proliferation in adult ovaries and testes. And the observations are there are 2 copies in the SM4, SM5, and SM6, and 1 copy in the SM1, SM2, and SM3. And there's a duplication of the gene in duplication of the XQ arm in PA4, PA5, and PA6. That's why you could see 3 copies of the gene. And one of the limitations for the analysis of mosaic condition is that they have used peripheral blood mucosal cell, buccal mucosal cells, and vaginal mucosal cells in SM1, SM2, and SM3, but they were not used for mutations in SM4, SM5, and SM6. This may be a limitation in identifying mosaic condition. And the other observation is that PA4, in the PA4, the PD had 2 copies of the genes for SMARCA1 and then BMP15, while it had a deletion for the GYG2 gene.
Okay, yeah, I know it's long, but this is the last experimental design. And my experimental design is basically a summary of all they were saying. So if you were you didn't know what they were saying, I will try my best to explain what they were saying earlier. So my research question is, could the occurrence of spontaneous monarchy be associated with mosaicism? So basically what they did, this is like a quick rundown, they gathered 40 patients with Turner syndrome, and then they classified them into, according to presence of spontaneous monarchy and primary amenorrhea. Next is they compared and compiled results they pinpointed for any consistent patterns and identified any mosaicism. So the first, it's a huge text box, but the first data we'll look at is that for under ACGH, the plus sign means that the first X chromosome is present, and the percentage or the number beside the plus sign are the percentage of cells that have the second X chromosome. So if we're going to look at SM2, this one, so it has the first X chromosome, and only 10% of the cells have the second X chromosome. Then next we will look at is the iFish results. The iFish revealed all SM patients and PA patients have mosaicism, even though that for PA patients 1, 2, 3, they have mosaicism, but it's very low. It's in a low degree. Next that we will look at, I just want to point out that the 2 N's for SM1 patients, those mean that they have a wild type sequence and there is duplication happening. So it's not that they have the second X chromosome, because according to ACGH, only less than 10% of the cells have the second X chromosome. Next that we will look at is the results for SM patients 4, 5, 6 for the 3 different genes that was tested. So a mean of 2 copies were identified by the RT-qPCR, which means that the second X chromosome can be found, which you can connect it for the ACGH, because for this one it's 30%, 75 and 75. So they used 30% as the threshold for the mosaicism detection. Next is those random 3 N's for PA5 and PA6. So all PA patients showed 1 copy of the 3 analyzed genes, except for PA5 and PA6. This is because of the duplication of the only XQ arm in more than 30% of the cells. Next is the 2N, and then there's an N, and then there's 2N for patient number 4 for primary amenorrhea. So PA4 showed an increment mosaicism levels only on these 2 genes, and a mosaicism deletion in the GYG2 gene. So in summary, we could conclude that mosaicism levels influences the occurrence of spontaneous menarche in syndrome. This is because if we're going to look at the ACGH results, all of them have the second X chromosome is absent, and as well as the rest. So if you're going to compare it for patients with spontaneous menarche, the second X chromosome is there, but they have a mosaicism. So the higher the percentage is, the more normal you are, even though you have Turner syndrome.
So in conclusion, as we've seen, using the ACGH, iFish, and RT-PCR, we have seen that there are different gene dosages and different genes in both Turner syndrome patients that are characterized as primary amenorrhea and spontaneous menarche. Therefore, this difference in gene dosage could contribute to the difference in ovarian function. And also, we have seen that there is different levels of 45X and 45X true monosomy and 45XX mosaicism in both Turner syndrome patients and patients with spontaneous menarche. And the most important thing from this is that we have also seen that there is a positive correlation between having an increased level of mosaicism and spontaneous menarche, which refers to developing normally, pubertally, in Turner syndrome patients.
So we looked at the significant limitations. So with the GH, we saw that looking at the chromosome, you can see that there's a duplication or a deletion in the chromosome, so that's a key. And also combining two different techniques. So for example, they combine IFISH and RT-PCR. So with that, that helps you to clarify your findings. And limitations, the sample size was small. There's only 40 patients. And the article did not state the ethnicities of the patients. So that, if they added the ethnicities to the research, that could have at least added some type of different conclusion to why these patients possibly have Turner syndrome and why they possibly have spontaneous menarche and primary amenorrhea. And then also the study did not include animal models. So with that, the research couldn't really compare the findings to see like whether this finding is accurate or if there's another way to basically clarify their conclusion. Lastly, having the detections of your CNVs will allow you to see that whether this disease is related to ovarian function. And these are our references, which include the research article, a supplement article, and our textbook.